So now it's my pleasure to introduce our Katz lecturer for 2025, Professor Dave Weitz. Professor Weitz is the Mallinckrodt Professor of Physics and of Applied Physics at Harvard, where he continues to enjoy an extraordinary career in the field of soft condensed matter physics, complex fluids, and microfluidics. Soft matter refers to complex fluids and materials that don't fit neatly into the more traditional classes of materials. Gels, foams, emulsions, everything you can't carry on and you carry on an airplane that's over 3 ounces. And essentially everything in our bodies, from membranes to cells to tissues, are all examples of soft matter. Dave Weitz is among the most outstanding soft matter scientists in the world, a scientist whose impact spans not only fundamental discovery, but also new techniques for studying soft matter and practical applications. Dave grew up in Canada, rumored to soon be our 51st state, and received his bachelor's degree in physics at the University of Waterloo. He earned his PhD in physics from Harvard in 1978, working with Mike Tinkham on superconductivity, after which he went to Exxon as a research scientist where he worked for almost 18 years. Among his early contributions at Exxon was his influential research in colloidal science, where, for example, he demonstrated that colloidal aggregates are fractal and revealed the dynamics of how they form. In 1988, Dave Weitz and Dave Pine invented an important dynamic light scattering technique called diffusing wave spectroscopy, where we learned yesterday that the DW does not stand for David Weitz. Before their work, dynamic light scattering could only be used to study transparent substances, but with diffusing wave spectroscopy, they extended light scattering to opaque materials such as paint, milk, shaving cream, or even human tissue. And I have a very distinct memory of sitting in a seminar at Boston University when I was in graduate school. It was probably 1990, 91, 92, watching Dave shine a red laser pointer through a glass of milk and showing, big speckle patterns on the wall, giving us an example of diffusing wave spectroscopy. And just like he was yesterday in his lecture, Dave is always so animated with so much passion for what he's doing, like just doing science for the love of science, that he became a hero of mine at that moment. In 1995, Dave joined the faculty at Penn and moved again to Harvard in 1999, where he began to explore the synergies between soft matter physics and biology. Using microrheological techniques, he studied the cytoskeleton of living cells and became a pioneer in the burgeoning new field of microfluidics. Some of Dave's contributions to microfluidics include methods of producing uniformly sized emulsion drops that improve consistency in manufacturing, and methods of making double emulsions, drops within a drop, for the controlled release of fluids and medications. Dave has trained legions, I couldn't get an exact count, of graduate students and postdocs, many of whom have gone on to their own distinguished careers, building upon the research they started under Dave's tutelage. He's published nearly 900 scientific papers, founded more than 20 startups, and created popular courses at Harvard, such as Science and Cooking, which has attracted many millions of online views. And I'm sure that you don't remember this, but Dave made a really big impression on me when I was a young researcher at National Institute of Standards and Technology in 1997 that I want to share with you. So we had submitted a paper for publication, I think to PhysRev Letter, and it was one of my first papers as like the lead author, not the first author, but like the group author. And very proud. I was certain that this was the best paper that had ever been written and submitted to PhysRev Letter. And we got the reviews back, and I was not happy. And I read those reviews and the critical comments in the reviews, and I got very defensive. And I thought, they don't think I know what I'm doing. And so I wrote a rebuttal letter. And I hadn't had a whole lot of experience writing rebuttal letters at that time. But I wrote a killer rebuttal letter. And I sent it in. And then, I don't know, a week or something later, I get a phone call in my office at NIST. And it's Dave Weitz. I'm like, what? Dave Weitz is calling me? And I don't remember exactly what you said, but it was something along the lines of, are you out of your mind? This is not how you write a rebuttal letter. And he talked me down from the ledge and went through how to write an effective rebuttal letter and how to understand that these are opportunities to make our papers better. They're not meant to be criticisms, although some reviewers are kind of jerks. But mostly, I mean, and honestly, I've never had a paper that did not benefit from the reviewing process. But that was a really important moment, because if you hadn't called me, I don't know, I could be flipping burgers right now because I pissed off so many reviewers that, you know, who knew? Anyway, thank you for that really important mentoring moment. So Dave's prolific academic and scientific contributions have been recognized by his election to the National Academy of Sciences, the National Academy of Engineering, and the American Academy of Arts and Sciences, and as a foreign member of the Chinese Academy of Chemistry. His distinguished career includes numerous other honors and awards, such as the 2024 Bauer Award and Prize for Achievement in Science from the Franklin Institute. And the citation for the Bauer Award reads, "For transforming our fundamental understanding of squishy materials ranging from gels, polymers, colloids, and emulsions to living organisms. His innovations led to the development of microdevices for applications ranging from rapid disease screening and drug discovery to cosmetic products. A master educator, he has popularized science through the physics of cooking," end quote. From the physics of foams to the creaminess of chocolate pudding to everything in between and beyond, Dave Weitz is a consummate scientist, educator, and entrepreneur whose career spans the entire spectrum from fundamental science to everyday phenomena to important practical applications. And it's because of his amazing achievements and his really beautiful science that Dave was chosen as our Katz lecturer for 2025 and by our students completely independently as the Students' Choice Seminar speaker this year. So we were delighted that we could combine them and that he could come here and now give us the Katz lecture. So without further ado, let's welcome Professor Weitz.
So with that introduction, I have nothing left to say.
I left you like 15 minutes.
I'm the only thing between you and food. Anyways, it's a pleasure to be here. I'm going to be, oh, there's a screen in front of me, good. I'm going to figure this out. Okay, I'm going to actually talk about a bunch of things that Sharon mentioned, both using microfluidics to make things, using microfluidics to do biology, and using microfluidics to detect things. I'll try and talk about each of them a little bit briefly, and I'll try and watch my watch so it doesn't go on too long, and I'll stop if it does. Not sure why this all came out quite so uneven, but that's because I transferred from my computer to somebody else's computer. Never mind. These are everything I tell you about is not really my work. I don't do any work anymore. It's the work in my group. And this is a group of people who've done it. I will not necessarily tell you who's done each thing, but all of it is done by really a wonderful group of talented students and postdocs, some of whom are listed here. I'm going to tell you about a bunch of different things, so I'll go a little bit quickly, and sort of a big room. But, you know, if you really don't understand me, jump up, wave your hands, and if I see you, I'll let you ask a question. I really don't mind. Okay, so microfluidics. You know, I don't know about you, but, you know, when I was a undergraduate student, this is not how I transferred fluids. Do you remember those pipettes? Oh, you're not supposed to do that, right? You're supposed to bulb. Okay, these are modern pipettes now. But those are old, right? Now when you do it with automated pipettes. You do this with these robots. This is pre-AI robots. But the thing about this is that's still an old way of handling fluids. I'm going to tell you about how to handle fluids and to work with fluids, not in wells like this. This is a 384 well plate. It works with one of these robots. This is probably several generations old. Each of these wells has maybe holds roughly, 10 to 100 microliters. That's not a whole lot, right? But we do things in small drops. So these are small drops of usually water, but doesn't have to be, of a fluid. And this is a reaction chamber, or this is something that we build something around. It's in an inert oil. The size of these, the scale of these are not microliters, but picoliters. So think of that. If I have 1,000 experiments and 1,000 picoliters, that's a microliter. That's less than one of these experiments. So there's 8 orders of magnitude difference in scale. And that's really where the power of microfluidics comes. And that's what I want to tell you about. You can make perfect drops. They can be, you can use them to contain things, and you can use them to build things around them. So the common phrase of this is called lab on a chip. Now, I'm super impressed by the building here because you have so much space. So as chair, Sharon never has to worry about, is there space for a new faculty member? But often places have no space for a faculty member. And so they come to you and say, well, you're a new faculty member. We'll give you a lab. We'll give you a lab on a chip. It's about this big. That's all you need. No, not really, but that's the idea. And I'm going to tell you about some of that. I'm going to tell you some applications that we use for doing biology. Some new things and some old things. I'll do that in a bit. But before that, I want to tell you about a different way of doing things. And that is, remember those perfectly round drops? The thing about a drop like that has surface tension. It keeps it in the shape of a sphere. And we can use that to do a whole bunch of other things. And I want to show you a little bit about that. So to do that, when you're at Harvard and you want to do microfluidics, you go across the street and you ask George Weitz, please, George, can you show us how to do soft lithography? And I'll show you some things with soft lithography. But it turns out that that's not the best way if you want to make materials, because materials often have all these different kinds of solvents, and the PDMS, the silicone rubber that you use for soft lithography, is not particularly good with materials or with solvents. So we wanted to do something that was super simple, super easy, but had a much more robust material. And of course, glass is the best material for chemistry. So we invented what we called capillary microfluidics. And literally all it is taking capillary tubes like this, and you can draw them out. You go to the biology department where they do patch clamping. If they know how to do this, they have machines. And you ask them, please, will you show me how to do that? And you can draw them out into these really narrow points. This is seen with a picture with an image with a camera. I'll show you an image with a microscope. But you can, there are machines that do perfectly, and you can do it absolutely reproducibly, and you can make them into a very, very narrow point. The inner diameter of one of these capillary tubes is about half a millimeter. You can draw it down to a micron diameter orifice really reproducibly and really easily. And then you want to align it. So what you do, this is a cylindrical capillary. And so if you fit a circle into a square, you take a square capillary and just thread the circle, the cylinder into it, and lay it against one of the corners. You have it perfectly aligned. So you can align these things really, really simply. And so you do that, and you make a device. You have the small 4 corners here where you can pass a fluid. You can pass the fluid through the middle. It's tapered down here. You hold it, I told you it had to be super simple, right? So you hold it together with the most important ingredient, 5-minute epoxy, and you make a device, and this is what it looks like. This is now seen with a microscope. You can see this is the narrow orifice, and we're making drops, and they look really perfect in size. In fact, one of my really wonderful early students who did this, he said, they're perfectly narrow in size. I said, well, how narrow are they? He says, well, about 3%. I said, oh, good. How did you figure that out? He said, well, this was a long time ago. We had this high-speed image camera taking images. And this is really before you had all these nice image analysis programs. He had one, and he fit this to a circle, and he said, I fit this to the circle, and I measure the size and measure the variation. I said, well, good. How accurate is your fitting? He said, about 3%. And how accurate is size? About 3%. I said, okay, you can't measure the difference in size. You just can't measure it. And these really are perfect. The size of these are a few micron, about 10 microns, so they hold a few picoliters in fluid. This should look familiar to you. It's going drip, 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 drip. If I increase the flow rate, what happens? You get a jet. If I try and hold my laser where it breaks off, you see it doesn't always break off at the same position. The size distribution is a little bit different. This is dripping, jetting, but still you make really nice drops. You just control the rate of flow. You can do this backwards. You can, instead of going in this direction, we call this a co-flow. You can counterflow. Then you have a flow focusing phenomenon. The outer fluid now focuses the first fluid. You get a more narrow flow. You get smaller drops. See, the flow is coming this way. The outer fluid's coming around here, going through here. You get a flow. You can get dripping. You can get jetting. You can barely see the orifice now. There's just a piece of dust here, but you get the same thing. The nice thing about this now is that, look, if I set it up that way, I have some extra space here and I can align things really well. So I can put a second drop, a second capillary here. I can align it perfectly. And now I get something that looks like this. One fluid, two fluids, and you get a drop inside of a drop. Maybe it's better if I show you this. Here I'm just changing the flow rate, and you can see you have a core in the middle and a shell, and the size of the shell compared to the core just depends on the flow rate. Again, very reproducible. It looks like it's going the wrong way here, but that's just a stroboscopic effect. They're just flowing downstream. Already here, there's something you can do interesting things. So I can take that shell and can polymerize it. In order to open it up, then I have to crack it. I crush it. I call these the Pac-Man structures. You crack them open. I can do what I call melt emulsification. I can use a low melting temperature oil on the outside, and I can heat it up into a fluid and then cool it down when after I make the structures. I have a core shell structure with a solid shell that's just solid, but I can easily liquefy it. So now if I put something inside here, I have some dye, here I have some particles, and I heat them up, you see what happens, they melt and they release. This becomes sort of interesting if I want to take a hydrophobic and a hydrophilic material, and this is something you often want to do when you want to deliver drugs. You want to deliver two different drugs. They work synergistically. One may be hydrophobic, one may be hydrophilic. This is a very easy way of doing it. This is not a drug, these are just dyes, a red on the inside, that's a hydrophilic dye, green on the outside. But now to release it, I just heat them up, and this can be done. They can heat them at body temperature, for example, or a little above body temperature. And you can see now you can deliver exact amounts in exactly the right region, exactly where you want. And we've used this for a variety of different active drug materials where we want to deliver the same amount or a fixed amount in the same place. Jörg Werner, when he worked with us, he used another polymer. He wanted to have something that was pH dependent. So he used this particular polymer. This is something that will expand and contract depending on the pH. He made capsules of that. He polymerized the shell. And so as you change the pH, you can change the radius. Here's going up and down in pH. You can see this. This is going to a higher pH. You get some crumpling as it goes up, but they expand and they contract. And now you have a hydrogel. You can expand it, contract it. You change the pore size of the hydrogel. You change whether it releases or something, so you can actually control the release just by changing the pH. So here's a step release just by changing the pH that it's in. You can also maybe do the opposite, open it up, have something diffuse in, and then close it and collect things that way. And Jörg made a whole bunch of different things where you could deliver over different times just by controlling the amount of the structure of the hydrogel. Here's something where he delivered over the course of one day or several days. And again, it's just by controlling the structure of the gel. The nice thing about this is if you're thinking about encapsulating something, making a core shell structure, you can encapsulate something, but remember how we make it. we make this kind of structure. This is just shown schematically. This is the outer fluid. This is the shell fluid, and this is the inner fluid. And the shell, the core and the outer fluid and the shell are three completely different materials, different flows. So if I want to encapsulate something, I can put it in the innermost flow, and I can encapsulate something really easily. And this is actually a very nice thing if I want to now think about trying to deliver things, if I want to think about trying to deliver drugs. And that's what I want to turn to in a bit of a discussion about how we think about something like that. So the first thing that we said, well, what's a really nice structure for drug delivery? Well, something like a vesicle is something that's been used for years as drug delivery. So one way of making, well, the common way of making a vesicle, so a vesicle is a bilayer of phospholipids folded around to make a shell. And I'll show you examples of that. And normally what you do to make these things is you dry phospholipids. You dry many layers of phospholipids on the surface and then bubble nitrogen and water or use electric fields. Somehow you make them into vesicles. And then you can shear them or something to make them smaller. And then you have to fill them because you have no way of filling them. Or you fill them, you put whatever you want to put on the inside everywhere, and then you have to separate what's inside the vesicles from the rest of the fluid. You have to try and recover it. It's really a pain. But now we can do that. We can fill them with 100% efficiency because the flows are completely different. They're completely separate. We can put what we want on the inside and just fill it with 100% efficiency. To do that, what we can do is we can make this into some solvent. We can make it a somewhat volatile solvent, evaporate it to get this bilayer structure. But ideally, we'd like to start with a much thinner shell. I won't show all the details, but we figured out a way, we had a postdoc who figured out a way adjusting the flow a little bit to make a very, very thin shell structure. And here's these structures where very, very thin shell. And now you can coat the inside and the outside with phospholipids or with diblock copolymers, some kind of amphiphilic molecule. They'll go to each interface and you get a bilayer structure. Here's where we're really trying to make a vesicle structure. So we're trying to make both the inside and the outside with phospholipids. The hydrophobic regions are on the inside. This is basically a membrane, just like a cell membrane. The hydrophilic regions are on the outside. And there's a small amount of this solvent that we have to evaporate. Here's some of the solvent that's shown. This is a series of images. And here's a vesicle that we made. This is the solvent that's slowly evaporating here, just small amounts. And it's just a small line tension that's pulling this together. And you can see that this really is a vesicle. This one you can't see because the contrast is not so good, but this is not bad. You can see the fluctuation. So this really is just a bilayer structure of phospholipids. So we can really make these large unilamellar vesicle structures. And we can use this, and Laura Arriaga used this to study the phase separation of different kinds of phospholipids on the surface. You can see here, it's undergoing phase separation. Here's another one. They've got just phase separation of phospholipids. Very nice way of studying the behavior of phospholipids on the surface of a membrane. Here's another series of phase separation using two different dyes. But can we do something different? And can we really do something that might make a difference? So the normal way of making all vesicles, all small vesicles, I call small vesicles if you make these large vesicles, what you can do is you can run them through a small, it's basically a narrow flow device that will break them into much smaller structures, still vesicle-like structures. When they're very small, they're typically called liposomes, and they're actually widely used and have been used for years for drug delivery. And this is the way a typical liposome will look, is have two leaflets, and they're made of the same type of phospholipid. But let's think about something a little different. Can we make something that has different kinds of phospholipids, different leaflets? It turns out that if you look at what cells are like, their membranes are like the membranes of a vesicle, but they typically have different leaflets. The components of the leaflets are different. And one structure that's becoming very interesting for delivery of drugs is to take pieces of the membrane of the cell. They're called exosomes. They're basically structures, vesicle-like structures from the cell, and they'll have completely different inner and outer leaflets. So can we do the same? Can we make an asymmetric structure? And the answer is yes, we can, but now instead of just making one drop inside of another drop, we make two drops inside of a third drop. One drop, a shell, and a second shell. I haven't shown you the device, but we can easily design microfluidic devices that do that. And then if we put one type of phospholipid on the inside and a second on the outside, they'll diffuse mainly to the nearest surface. Okay, it won't be 100%, but it'll mainly be to the nearest surface. And if we use different phospholipids, we can get this asymmetric structure. And so we did this. How do we test it? Well, we can put something that will bind a dye molecule on the inside or the outside, and then we can expose it to dye molecules. If the binding is on the outside, you'll see the outside light up. If it's on the inside, it won't, if you wash away the dye. And so here's an example where we did this. You can see here, it's on the outside, and you can see it light up. If you look carefully, if I turn the lights down, you'd be able to see a little bit here. So there's a little bit of crosstalk. There's a little bit of diffusion across it, but it's really pretty good. It's pretty asymmetric. And you can do this a lot of different ways. You don't have to use just two types of phospholipids. You can use a diblock copolymer, some other kind of amphiphilic structure on one side, and something on the other side. And Tina Wong made a whole bunch of these things. She made these diblock structures, these asymmetric polymer lipid structures. And she was interested in doing this because then you can look at the mechanical properties, and you see that they have very, very different mechanical properties. It's not so surprising if you look at something that has just phospholipids. It's a very, very soft, very flexible structure. And the way you measure it is you take a pipette and you suck the structure in, and you measure what kind of pressure it takes to deform the structure. This is called micropipette aspiration. I don't need to tell you the details of it, but this is basically a force displacement curve. This is the tension that you pull it in and how much you strain it. And this, if you look here, this is a relatively low tension and is very linear. This is the phospholipid. And as you add more and more diblock copolymer, you get different kinds of structures. And this is all then a very nice kind of study because you can make things that are completely uniform, with one layer of one thing, another layer of another things, or you can make things that are mixed so that they have patches of polymer and patches of phospholipids, and you see all different kinds of mechanical structures. So you can see this really nicely. But if you really want to use these things for drug delivery, then you want to make them much smaller, and you want to keep them, in some sense, very simpler. Much simpler. And this actually is something that we got started thinking about this right after the pandemic. So I don't know where you guys were during the pandemic. I was in Boston. I remember, I don't know, a few months earlier than now, in 2020 or something, they came and shut my lab down. And we couldn't do anything for a few months. We finally got back to the lab. It was a terrible time, but really, 1 year later, I remember I was sitting on my computer online, trying to get a line, logging in at midnight, trying to get in line to get a vaccine that had been developed and perfected in less than a year. And these are these mRNA vaccines. I was in Boston, so it was, of course, Moderna vaccine. And there were a lot of things that were essential for creating the vaccine, one of which was the delivery mechanism, which were these lipid nanoparticles. And that got everybody really excited about lipid nanoparticles. I don't want to go into the details of lipid nanoparticles. They're like liposomes, but they're not quite like liposomes. They work, as we saw, remarkably well for mRNA. The trouble is now people start asking, well, can they work for other therapeutics? And the next generation therapeutics are not mRNA, but they're going to be large proteins such as CRISPR or Cas9 that will actually do gene editing. There, you'd rather not deliver mRNA and express the protein in the cell, but you'd rather deliver the protein itself. And now I have friends who are trying to take these new molecules and make them smaller. You can't use the LNPs. They don't work for the proteins. You try and use viruses. Viruses are not that good, and the capacity is not that big. So you try and engineer the Cas9 to make it, or the CRISPR to make them smaller. That's not really that good. What you'd really like is something that will allow you to deliver these large proteins. And so we said, well, let's go back to these asymmetric structures and see if they'll work. They're exactly what you need. And rather than do it with the traditional microfluidics, we turned to a technique that we had invented like 25 years ago, which is really a very simple technique. You make an emulsion of a water in oil where you have one phospholipid as a surfactant, and you pass it through a second phase through an interface where there's coated with a second type of phospholipid, and you can just pass it through, and you end up with an asymmetric structure. And it didn't work so well when we first did it, but we were trying to do 10, 20 micron diameter particles. And if you think about trying to pass something that's 20 microns through an interface and have the interface curve around it, it's tough. But if you use something much smaller, it seems to work much better. And so we went down to 100 nanometers, and now it works really pretty well. Here's the structure that we get. This is the distribution. We can get them very small, and we can pass it through the second interface, and we can really get very nice structures. And we did a lot of study about understanding things. It turns out that it's not 100% efficient, that we lose some of the vesicles, but depending on the surface tension, we can do much better. If the surface tension is really low, we can do really well. So there's a lot that we can do to optimize. We understand how it improves it, but this is a really simple way to make these asymmetric vesicles in a way that we have whatever we want already inside it. So we want to put CRISPR molecules inside, we can. We can do the same kind of assay. We can do the same test to show that it's asymmetric just by quenching by putting dye on the outside and measuring that. We do that. It's really asymmetric. And now we can just test these things. Are they efficient? Well, the goal of the lipid nanoparticles is to get something in the inside of the carrier into the cell. Will these work the same way? And so here's just an example where we use fluorescence to label these structures, and you can see that they do get taken up in the cell. They get taken up by endosomal process. Basically, the cell will wrap a membrane around it, and it'll go inside of the cell. That's what you want. And if they're asymmetric, they seem to get taken up much better. So I won't go through the details, but if you look carefully, you see that the asymmetric ones here, these are two layers that are asymmetric, they get uptaken by the cell much more efficiently. Why is that? I don't really know. I can speculate. We always see that. I speculate that the membrane of the structure looks more similar to the membrane of the cell. So we can adjust the charge to be more similar to the charge on the cell or different to the charge. We have a lot of control of that, but it seems to work pretty well. Then we have to ask not only if they're taken up by forming an endosome, by the membrane wrapping around and bring it in, does the molecule on the inside get released? So can we release it inside? We can do that by taking something that will express fluorophore in the cell. So we put basically an mRNA for GFP inside the asymmetric vesicle and ask, do we express GFP on the inside? And the answer is yes, we do that very, very well. And if they're asymmetric, they seem to get both taken up and released much better. Now, again, this is not we haven't tried to optimize this, or we've tried, but we haven't fully optimized it yet. We can control how to make it release because we have control of what we can put on the inside and what we can put on the outside. Also, it's interesting that the cells are much happier when they're seeing asymmetric structures. Again, I think it's because they're seeing something closer to what they're used to. And so the number of cells that are killed by these asymmetric structures goes way down, so they're much safer. We can do a lot more. We can coat these normally with in the way that these structures are normally used, liposomes are normally used. They're coated by PEG, and that's to make them what's called stealth, so that they're not taken up by the immune system. They can flow through the body. We have tried to coat them with things that will target different regions. What we think is that that putting polysaccharides of different types on the surface might help target different cells. So we have simple ways of putting polysaccharides. Basically, we bind a polysaccharide to a cholesterol. The cholesterol goes in the membrane, and we can coat the structure with polysaccharides. And we find, again, that seems to help the uptake. I'm not sure that it targets things yet, but we're going to try that, whether we can target other than the liver, whether we can target different types of cells by changing the polysaccharides, but we hope that we can. And the bottom line is that if we ask, can we improve things, we find that if we use polysaccharide-coated structures, the uptake is better, the transfection is better. If we use asymmetric coated structures, that's also better. So this combination really does seem to improve and does seem to solve some of the fundamental problems with LNP. Will it actually work? I don't know yet. It's promising enough that a former postdoc that I knew who was in Boston, he now has a company that's trying to deliver CRISPR molecules. He's agreed, he's interested enough, he's having such problems delivering them that he's actually going to take these things, we're making some for him, and he's going to actually do some mouse model tests to see how well they work. So stay tuned, I'm hopeful. Okay, I should talk a little bit about some commercialization ideas. This is an old commercialization. It's sort of a fun commercialization. You know, people would come to me and say, you're making these materials, you're making these drops, they're beautiful, but you're making them one by one by one. Can you possibly commercialize something? Or can you possibly make enough to be reasonable? And the way I say, if I want to make a reasonable amount, if I can commercialize it, then I obviously can make a reasonable amount. So I wanted to do something, and you know, we've been working on drug delivery forever, and we're still doing it. We're still nowhere close to actually trying to commercialize something. So that's the wrong thing to actually prove that this can be commercialized. I have to find something else where there's an emulsion-like structure that people will pay a lot for, because it's not free. It takes a lot of, it's costly to make it this way. I have to find something that people will pay a lot for. So what is something that people pay a lot for that's an emulsion? Well, it's cosmetic. So this should be here. I think well, never mind. I won't explain why it's happening. But anyways, cosmetics. So we started a little company called Capsum, and we made these microfluidic structures. This is the first thing that we commercialized. So if there's anybody here who's from Korea, they will know right away that Amori Pacific is one of the big cosmetic companies in Korea. I guess I learned a lot about cosmetics during this exercise. It's a big company in cosmetics. They commercialize this. But then we made other kinds of structures. Here's one kind of structure. This is actually a commercial operation making a core shell structure. Let's see. Let me go here. This is a core shell structure. This is it was marketed by, so La Prairie, who knows what La Prairie is? A few people. Yes, you do, me too. It's a high-end Swiss brand cosmetic. People will pay a lot for this, right? This is the Skin Caviar Liquid Lift, right? These are the microfluidics. They pay a lot for this. Is I think this is about 50 mil, $500. Okay, high-end, right? So here's another device that we made. This really is the capillary device. We're making things, and with this we made this product, Hydra Beauty Microserum. I recommend that you go and you look this up, because they still have on their website, they have something that shows that it's made with microfluidics. The first 9 months, they sold 1 million units of this. In fact, Chanel liked it so much that they just bought the company. So they bought Katz. And I can tell you they paid less for the company than they paid for the advertising to introduce this product. So now you know what the cost of, or what the advantage of the high-end cosmetics. So this at least proves that you can really scale things, and you can do things at a scale even when you're making things one at a time. And you saw the way you make it. You just have lots of them. They were making drop by drop by drop, but you make lots of them. So now let me spend a few minutes and show you the other side of microfluidics. Here, up to now, I've showed you how to use this. And what we're really most interested in is trying to do it for encapsulation and release for drug delivery. But if we want to use this as a reactor to do experiments in, so let's go back and think about it. A 1 micron drop that has a volume of about 10 to the minus 15 liters, a femtoliter. Things that you're sort of aware of things like that. If you look at a nucleus of a cell, it's about a micron in diameter, a couple microns in diameter, few femtoliters. What's really interesting about that is that you can put 1 molecule in a femtoliter, and you're already at nanomolar concentrations. Nanomolar concentrations are the concentration where a good catalyst starts to be quite active. And so that's really why you encapsulate things, is you have a small volume, but you can still have a larger, much larger concentration. 10 microns, that's our sort of sweet spot. That's about a picoliter. 100 microns is a nanoliter. And we make these things at about 1 kilohertz. So if we want to do experiments in them, we should design structures that allow us to do the experiments. And the nice thing is that we can do lots and lots of experiments with very, very small total amount of volumes. And we can improve the signal to noise, not because we improve the signal. The signal is usually limited by whatever is creating the signal, but we reduce the noise because we have the volume much smaller, so the signal to noise goes way up. So to do this, we designed a whole bunch of different microfluidic chips. Now we go back to the George Whiteside's soft lithography chips using PDMS. So we designed a whole bunch of different chips where we make the drops. We can split them into 2 parts, into 2 different parts. We can split them into large and small. We can take 1 drop and split it many times, so we can have a large drop and make it into very uniform, smaller drops. We were trying to do something really crazy. We were trying to understand how colonies or consortia of microbes work. So we wanted to build a chemostat. So we designed a chip where we take a small amount off. We can then use that to analyze, and we add something so we can keep the concentration and the level of nutrients the same. So we can extract something. We made a chip where we can mix things. If you want to mix things, it's actually very difficult to add something. So we take 2 drops with reactant A, reactant B, and we merge them. Or we invented something we called a pico-injector, and this literally is injecting a picoliter of fluid or a small amount of fluid into each picoliter drop. You can see this is a dye just to show that you can do it. These are electrodes. So there's an interface. You have to break the interface when it goes by. You just do that by switching on an electric field. But you can control that, so you can have something where you can have, here's 3 pico injectors. Only this one is active, but here they're different colors. We can switch them on a drop-by-drop basis, so we can add, mix and match. We can add whatever we want. You can detect it. We use fluorescence. These are all going at 1,000 drops per second of that order. And then if you really want to do something, you want to say, well, I want one of these drops and not the others. So you can sort it, so you can select it. This is a sorter that we use to select these drops. So we made all these devices. Let me show you a few uses, both some old ones and some new ones. So the first one that we did was we tried to improve an enzyme. We did something. When we did this, it was sort of a very simple thing to do. We use horseradish peroxidase because that's something that will operate on a substrate and make it into a dye, make it fluoresce, or luminesce, so it's easy to detect. It was probably not the best thing to do because who cares about improving horseradish peroxidase, but we wanted to show that we could do that. So you do that by not trying to do the chemistry on the enzyme, but rather by doing what's called directed evolution. This is using what the standard way of using evolution, but this is something that Frances Arnold took the standard way of evolution, but tried to do it directed, and she won a Nobel Prize for this. Basically, you make a library of mutations of the genes that code for the enzyme. You make a library of mutants, and you express each of the mutants, and you ask, okay, does this mutant work better or worse? And you pick the ones that work better. And so we did this, but this was quite a few years ago where we weren't able to express them that easily. So we worked with Dane Wittrup at MIT, and we expressed them on the surface of yeast. So we put the gene. We made a plasmid of the gene, we put it in yeast, we expressed the enzyme and displayed it on the surface, and we encapsulated the yeast cell in a drop. And so this is all the drops that you can see that have yeast cells now fluoresce because the enzyme is operating on the substrate and it's causing fluorescence, and it just encapsulates the fluorescence. So now the experiment is pretty easy. You take a library of mutant genes, you make them into plasmids. You put one plasmid per yeast cell. You put one yeast cell per drop. You just ask, does the enzyme express something? Can you see it? If it does, you sort it out. The nice thing, of course, is when you sort it out, because they're yeast cells, they're living, you can grow them up and you can do another sort. So you can do several rounds of sorting to really get exactly the ones you want. And so we did all these experiments, and we looked. This is just measuring the average fluorescence, the average level of fluorescence. If we use the wild type, you see some average. At first, the fluorescence, when you look at it, most of them are no good. They don't work. So the average level is really low. But after a while, after a few rounds of sorting, round 1, round 2, round 3, you get so that you're choosing things that are a lot better. So now we can go and look at these things. There's one yeast per cell. We can take them out. We can grow them up into colonies. We can sequence the colonies. We can see what we have. And we found that we started with a library size of something around 10 to the 7, and we found about 100 different variants, an estimate, 100 different variants where we could sequence them. And this is just the activity level. This is the wild type. Some were lower, some were higher. So we improved it to some extent. And we could go, since we sequenced them, we could see where the mutations are, and they're all over the place. The active site is here, and the mutations are all over the site, all over the place. And we've improved it to some extent, but then what we did was something else. We took the best 20. We probably should have taken all of 100. We took the best 20, and we did a second round of mutation. We took the genes that coded for that. We had them in the yeast. We took the genes, we mutated them, we made a whole new set of libraries, and we did a second round of sorting. And now, right away, we found things that worked. They worked a lot better. They worked so much better that we really couldn't even measure how well they worked because we were diffusion limited to measure the activity. But we improved things really substantially. So what are we doing? We're doing what's called a neutral selection. So this is a fitness landscape. A fitness landscape is, if you know what an energy landscape is you want to go to the minimum energy. Fitness landscape is just turn your head upside down. Fitness landscape, you want to go to the highest fitness. You want to go to the top of a peak. Nature has made something here. We want to go here. But in between, this is fitness space. So these are mutation space. In between, things work really poorly. So we want to get here. And jumping from here to here is really difficult. You have to find really one mutation or one set of mutations that get there. That's very, very difficult to do. So what we did instead was we found a whole bunch of ones that worked roughly as well. And then we found something that worked like this. And this is called neutral selection. It's difficult to do because remember, we had a library of 10 to the 7, and we only found 100. So it was 1 part in 10 to the 5. You're looking at 1 part in 10 to the 5. That's really hard to do. You don't want to do that. But with the drops, you can. You could actually do this using those robots that I showed you. Let me show you why you wouldn't. If you did it with a robot, you'd use about 5,000 liters to do the experiment. Here we use about 150 microliters. It would take you 2 years. I calculate, do this, 24/7, number of times, 2 years. Took us about 7 hours to do the sorting. I calculated this before the inflation of the pandemic and the recent inflation, and the cost then was 16 million. I know it's 20 million now. It cost us $2.50. That's why we could do this. But okay, this works, but still, if you think about it, finding one mutation in 10 to the 7, if you think of what the size of sequence space and the number of mutations you really like, it's still not that good. You'd like to do a lot better. And so we realized 2 things. First of all, we realized that we're doing the expression in living cells, and so we'll put 1 cell per drop. Why not just do a cell-free expression and put 1 template per drop and just do it cell-free? But then we realized, why do you put 1 template per drop? You don't have to. If you're doing cell-free, you can put as many templates as you want. Why not put lots and lots of templates in each drop? So you put many, many templates, many, many mutants in each drop. If the hit rate is very low, then if you have a hit, you know there's one of those templates is really still works. You just don't know which one it is. That's okay. All you have to do is sort that dropout, amplify everything that's in there, and do another sort, and just keep sorting until you do this. And so we designed something that would do this. We injected things. It turns out to be a rather complicated thing. We injected things. We added many, here what we're doing is actually we're injecting the substrate. We did the expression, then we injected the substrate. We look, and here's just an example where you can see that this is 1 wild type gene per drop on average. So you can see, per 10 drops, 1 in 10. So you can see a few of them light up. This is 1 wild type gene per 10 drops. And in each drop, there's also 10 to the 5 inactive genes. And again, you see the same thing. You can see a few that are really light up. So all you have to do is sort those and then dilute them, amplify them, and sort them again. Do several rounds of sorting. Okay, these movies don't play so well. Sorry. If we do this now, we can see something of the order of 10 to the 12 variants. Why is that interesting? If you can probe 10 to the 12 variants, now you can look for instances where the mutations can have 4 or 5 or 6 or 7 mutants and still find something that's active. And we've been able to do that so we can look way far away and find something that really does something completely new because we can add many, many mutations. You couldn't do that with any other technique. Okay, so that's something. I want to show you one last thing and just take a couple of minutes. Again, taking advantage of the compartmentalization, putting something inside, taking advantage of being able to put small templates in. I want to show you a way of doing really super sensitive detection. Again, this is motivated in part by the pandemic. So during the pandemic, I don't know about you guys, but I had to get tested. When I started going to work, I was allowed to go to work about two months after they closed my lab. They opened it up, but you were allowed to go to work, but you had to get tested for COVID. The students had to get tested every day. I had to get tested at least twice a week. And the way we tested then is with PCR. That's what we did. Gold standard for testing very sensitive amounts of material. In that case, it's a pathogen, a virus, but any kind of DNA you can detect with testing. It's the gold standard way of doing it. In fact, Harvard was able to do that so cheaply because another friend of mine invented something where we had these nose swabs, you had these Q-tips, you put that in, you put the Q-tip into you swab your nose, you put it into the reagent, and you sent that for PCR. He did something where the the swab was dissolvable in the reagent, and so they could use a machine to unscrew it. And that sped things up so much that it cut the costs of the PCR test. I think it cost them $15 a test, which was really pretty good. But that's really the gold standard of the test. The problem is you can test for COVID, you can test for the flu, but you can't test for very much. If you want to test for many things, what you do is you use sequencing. So if you have a lot of biomarkers that you want to test for, what you do is you try and sequence it. And that's very, very good. You can get enormous amount of data. The problem is you're looking for the needle in the haystack. You're looking just for a few things. So you're throwing away 99.9% of your data. It's very expensive. It's very slow. And it's not really that sensitive. So it has the advantage of being able to detect many things, the disadvantage of not being the easy way of doing it. So we asked ourselves, could we find something that would work and do both? And so the way we did this was we designed a test using PCR. We do what's called digital PCR. Digital PCR is nothing more than having a yes-no answer, diluting and compartmentalizing so that the template is in drops at a level of 0 or 1, digital, 0 or 1. And then if you want to know how much there is, you just count the number of positive drops. You do PCR. If there's something there, you get amplification. If there isn't, you don't get amplification. So that's very easy. It's very, very sensitive. It's much more sensitive than normal qPCR. I won't go through the details, but you can get super high sensitivity that way. But the trouble is, again, that it's just doing fluorescence. You're just reading what's there. So we said, could we find another way that we could use lots of different primers, still one template per drop, get amplification, but if we have lots of them, we don't know what it is. Can we find a different way of reading it out? And so we realized that a unique thing about DNA is that you can melt it. Right? You can dissociate it. You can go from double-stranded to single-stranded just by heating up the temperature. And the type of, the different bases melt at different temperatures. And so there's a melting curve that if you actually measure how it's melting, how it's coming apart, and you can do this with fluorescence techniques, you can measure exactly how it melts. And the temperature that it melts at is unique to what you're looking for. So you can use that as an immediate probe, but you can also do other things. You can put other regions on the primers that have melting temperatures way away from the oligo that you're looking for. You can put something at low temperature, something at high temperature, and you can adjust exactly what the temperature is. So you can put a barcode of melting temperature. And you can go even farther. You can put a probe. You can take a region that melts at low temperature, a region that melts at high temperature, and you can add some probe, some fret pair that will bind and will not will fluoresce because you separate the fret pair, and at some absolutely well-defined temperature will unbind and not fluoresce. So now you have another way of adding barcodes. And if you do this just by using melting temperature, you can add, here, my student who did this by combining two different colors, high and low temperature, she thought she could get about 3,000. This is when she prepared the slide. I think we're up to about 500,000 unique melting temperatures. So now we have exactly what the sequencing does. You have a barcode, but we're barcoding the PCR. So we're doing just PCR, but we can do a highly barcode. Here's just an example. These are the melting temperature of the one of the regions. You can see just by looking at different temperatures, these are the barcodes. You can distinguish them. This is actually looking at the fluorescence. It's not the derivative of the fluorescence, but it's not so important. Here it is again. And here's an example where using two different colors, in this case, I think there's 64, but I think Xing has gone up to about 180 different barcodes just as proof of principle. But there's no question, just looking at this, that you can go much, much higher. So now we have a way of doing exactly in the middle, doing both. You have the sensitivity of PCR, because this is just as sensitive, the inexpensiveness, the high speed ability, but you can do as many as you can easily with sequencing. Now, I'm sort of excited by that because this is a way of detecting lots of different things. I also see that the future of public health screening is going to be doing sequencing. You get your blood sequence to look for different diseases, but that's so expensive. Even a rich country like us is not going to pay $1,000 every year, which is what it costs now, for every person to get that test. But if you can do it with PCR, it's $20, $50. That becomes affordable. It becomes affordable also for the third world. We can do other things just quickly. You can do the same thing to detect proteins. Now you use pairs of antibodies. You use extension PCR. You have two different pieces of DNA. When the pairs of antibodies bind to a protein, you do PCR of the extension of the two pieces of DNA, and you detect this, what it is using melting temperature barcodes. Here's just an example where we're screening for a bunch of different proteins. These are just common proteins, but we can get down to sensitivities well below physiological levels and do many kinds of sensitivities. So again, I can show you a whole bunch of other things. Anytime you can do PCR, you can do multiplex this. You can do multiplex it just using these barcodes. Okay, so I've told you a bunch of different applications. All of them depend on the ability to compartmentalize, to make these small compartments that you can do very easily using droplets. You can do other ways. I'm not particular if you want to do it for wells or whatever. It's just the whole idea is compartmentalizing, and that's what the microfluidics really allows you to do. And so with that, let me stop. So, sorry for the slides not working exactly well, but if there's time, you can ask questions, but thanks for your attention.
Thanks very much, Dave. I just want to know, is there anything you cannot do with microfluidics and drops? We have time for questions, and Sheila is going to run the microphone around.
You can't make a great dinner that we're going to have now. Yet. So if you don't ask questions, I get to ask questions.
It was a beautiful talk. I was interested in the asymmetry of the vesicles you can make with the outer and inner composition. I wonder what kind of physical properties result from that? So can you destabilize or stabilize, or could you just comment on the physics of the asymmetric membrane like that?
I mean, it's all open to you, right? You can change the charge. You can have one charge on the outside. You have a different charge on the inside. You can have them both charged the same way. You can put proteins that bind to the inner leaflet that will help release for the endosomal release. After it's released, you can put proteins on the outside or antibodies on the outside or something on the outside that will help target, not on the inside. So the flexibility is really there. Why the asymmetric structure works better, I can't really tell you. I don't really understand that. It just does. Everything that we do experimentally. It just seems to work better, at least in cell-based studies. We're going to try animal studies next.
So thanks for the talk. I'm curious about the commercialization. So especially when preparing a story for a paper versus preparing a story for investors. Do you have any rules of thumb for what constitutes a good finished product for each one and how to adapt from one to the other?
I can only give you my experience. My experience is, well, first of all, why do I commercialize? I commercialize for two reasons. One is if it gets to be something that the amount of work and engineering it takes is not suitable for a thesis, then it has to need an engineering team. And why do I do it? I do it because I think it's something that my goal is to have impact. It's not to publish another paper. It's not to publish some fancy paper. It's not to learn how to deal with these stupid reviewers who don't understand so much. I hate that, but no, I want to publish papers. I really do. And my students want to publish papers. But if you want to have a real impact, you want society to use it. And I'll tell you something. we're learning more and more. Society, they don't care about universities. They don't care about papers. They do care about jobs. They do care about healthcare. They do care about something that will impact them. And I want to do that. I want to have impact for them. And the only way that I know how to do that is to do it in a way that's commercially sustainable. It has to be sustainable, and it has to be commercially sustainable. And then the biggest difficulty, and the thing that I always fail at, because I fail many more times than I succeed, is finding the right business people. Science people, I know that a dime a dozen. I understand science. I understand technology. I understand engineering. But business, I try, but I always make mistakes. I'm not a good business person. And finding the really good business people, that's the difference between success and failure, is always having the really right business people. And, I have a day job, so I don't want to do it myself. But some of my students, they go out and they learn how to do it. I don't think they can do it necessarily from day one, but they can learn how to do it. And all the power to them. I know if startup companies had existed when I graduated, that's sure as hell what I would have done.
Great talk. Question about the liposomes with the asymmetric leaflets and targeting. So I understand that, for example, mRNA targeting, it tends to get into not only the lymph system, but it can get into other organs. Like there's rare cases of cardioditis and other things, but in general, also for other kinds of drug delivery, I'm wondering if you can think of droplet methods that could accelerate the discovery of either saccharides or compositions of the lipids that would target the cells more specifically.
I mean, there exist ways. The person I know who did it is Dan Anderson, and I would use what he does, and I think it's a brilliant way. What he does is basically take a whole bunch of different lipids and puts a sequencing barcode on it, and a fluorophore, and he injects lots and lots of lipids, all different, into one animal, and then he just looks, where do they go, and what are they? He sequences to figure out what they are, and that is a brilliant way in my mind of doing it. So I think that's the way I would go about trying to optimize the structure and the source of the lipids to do to improve the delivery. And if you think about it, you can put lots of barcodes, because it's sequencing barcodes. You can put lots of barcodes, so you can do a huge number if you can make them, and it's not so hard to make them.
Hello, and thanks for the great talk. I have a question regarding the extract microfluidic that you spoke about. It was very wonderful. I want to know, how did you do the extract? Is this based on the size or the size of the tube that you have in the besides? Is it possible to have multiple size of the extract at the same time, or can it based on the surface modification or not?
Yeah, I'm not sure what you mean by the extract, but...
The extract microfluidic that you have on the corner of the slides, on the bottom.
Oh, the extruder. Sorry, the extruder. Is that what you mean? We made these small structures?
You have a bulk, large tubes, and you have a small tubes at the corner that you extract microbubble out of the bulk.
The answer is, I haven't shown you nearly the range of different kinds of devices that we've made. And so I can't do much, but my students and postdocs can do all kinds of things. And so I've yet to see something that if we really wanted to do, we couldn't figure out a way of doing it. So there are a lot of more complicated, sophisticated structures that we can make a lot of different sizes, different coatings. So I think we can do a lot of that. If I could just answer a slightly different question, what I'm also observing, and I heard a lot of people here talk about the same thing. My students now tell me, well, we do all this work, but nobody can reproduce it because nobody can do microfluidics. And so can we do it without microfluidics? And a former postdoc from our group, Adam Abate, has developed a lot of ways of doing things without using microfluidics. And so we use it for a bit, but then we try and also do things without necessarily the complexity of microfluidics. And there's all kinds of ways of doing that. So I would say it's a long-winded answer to say that there's still a lot of development that we are doing and that can be done to make things easier, to make things more flexible, and to try different things. And I apologize if I didn't get exactly what you want to do, but if you come and ask me, I'll try and figure it out.
Okay, I will come later on. Yeah, thank you.
Thank you. A perfect time to do that would be right now as we break to have a short reception with some drinks and past appetizers, and then we'll reconvene in like 20 minutes or something for dinner. Let's thank Dave again for a beautiful lecture.